Hi everybody, and welcome to our third webinar from Abberior. My name is Julia Menzel. I'm an application specialist at Abberior, and I'm your host for today's session. And today we have a really cool topic. It was actually chosen by our audience in an online survey before, and you decided for live cell STED microscopy and appropriate labeling techniques. And I'm very happy that I have two experts here today with me, Francesca Bottanelli and my colleague Florian Grimm. Both will give a presentation and afterwards answer your questions here. And we will start with Francesca. I'm very happy that we have Francesca here with us today. And Francesca is an associate professor at the University in Berlin. And she developed her passion for cell biology during her PhD at the University of Leeds. And afterwards, she moved to the US, where she started to develop novel labeling techniques for live cell STED microscopy. And afterwards, she combined her both passions of cell biology and microscopy and works now with her own group on the Golgi apparatus and how proteins are traveling inside the cell. And many of her research questions she answers with live cell STED microscopy. And before I hand over to Francesca, I would like to remind you to use the chat function during the talk to ask your questions. And with this, I hand over to Francesca. Francesca, we are looking forward to your talk.
Thank you very much for the introduction. I am gonna take over presentation. Okay. And now you should be able to see my screen, correct? Yeah. Please let me know if there is anything that doesn't run or stops or anything. So I'll start my presentation. Welcome everybody, and thanks to Abberior for inviting me back for the second year to talk about our approach to understand cellular logistic using live cell STED nanoscopy. And I really hope in the next 20-25 minutes I will really convince you that live cell STED has been a very powerful tool to help us understand how proteins are transported around in cells and cellular membrane organization. Okay, I am not going to go into much detail about the biology, but just a brief introduction. So we want to understand how proteins are sorted through the secretory pathway in mammalian cells. And so cells are compartmentalized into different organelles and membranes with different function. For example, our main lobe is for the Golgi apparatus. The Golgi apparatus is often regarded as the post office of the cell because it's the organelle that is responsible for sorting into the endolysosomal system for degradation, to the plasma membrane for secretion, and back to endoplasmic reticulum, where all secretory proteins are actually synthesized. So, and while this is just a schematic representation of a cell, let me put a laser pointer on. While this is just a schematic representation of what's going on in the cell, all these intracellular transport steps actually occur over extremely short distances. So this is the textbook electron micrograph, and this is what the Golgi looks like. It's this stack of flattened cisterna, and if this is the Golgi here, and this is the endoplasmic reticulum, and this is a 1 micron scale bar, if a vesicle needs to bud from the Golgi and then fuse with the ER, it will only need to travel a few hundred nanometers. And that's one of the reasons of why we need super resolution microscopy to look at these processes. If you image the Golgi with a standard confocal diffraction limited light microscopy, you won't see much detail, like shown in this example image here, where we have stained a type of vesicle, which is called COP1 vesicle, which mediate Golgi to ER transport. But when you turn on the STED laser, that's when you really start seeing details. For example, this vesicular structure here, which we know are vesicles that transport cargo, shuttle cargo between these two compartments. So I'm pretty sure that for this audience, I don't really need to introduce STED microscopy. So let's dive straight in. So I divided my talk in two parts. In the first part, I'm going to talk to you about how we do dual color lifestyle STED imaging using SNAP and halo tags. And then in the second part, I want to show you some examples of how we're harnessing lifestyle STED, in particular in combination with gene editing with the CRISPR-Cas9 technology, to understand the mechanism of membrane trafficking and also membrane organization. And people can do STED microscopy with green fluorescent proteins. For example, GFP and YFP have been used for dual color lifestyle STED. However, we like to use organic dyes because they're much brighter and they're much more photostable. And to be able to label our proteins of interest with organic dyes, we use SNAP and halo. So SNAP and halos are not per se fluorescents, but they're enzymes. So they react with a ligand. It's a benzoguanine ligand in the case of the SNAP and a chloroalkane ligand in the case of the halo. And basically then our ligand will be covalently attached to the SNAP and halo tag themselves. And when this ligand is conjugated to an organic dye, to a fluorophore, our fluorophore will also be covalently attached to SNAP and halo. And because they have specificity for different ligands, they are the perfect set of tags for dual color live cell imaging. And to do STED microscopy, our favorite depletion laser, like many of us, I think, is the 775 STED depletion laser. And to perform dual color microscopy, what we use a dye in the range. This is just an example of the dye that we first use as the proof of principle, but you can use a dye in the range between 560 and 590 nanometer, and together with a dye that gets excited around, around 640, 650 nanometers. So these two dyes are pretty close in spectra, but they're sufficiently separated so that you can, spectrally separate them. And particularly, what's very important, you can hit them both with the same 775 nanometer STED laser. And that's why we can do quasi-simultaneous imaging. So back in 2012, we published a proof of principle paper, and back then we used as a far red dye, we used silicon rhodamine. I think that's still the golden standard for far red dyes for live-cell STED microscopy, SIR, and further derivatives that were then developed based on the original SIR, together with 590 dye. In that case, that was up to 590. And however, there has been actually quite a bit of development in terms of dyes, and there is a lot of combination of 640 and far red and red dyes that can be used in combination to perform dual color live cell STED experiments. And we actually wrote a review last year about this, about development in live cell STED microscopy, and this is actually not a complete list because a few months after we published our review, actually a few very important papers came out even further developing from these dyes. And what about more than two colors? This is actually being done. And to do 3-color live cell STED, Stefan Hell's lab, for example, has used a red and a far red dye together with a long stalk shift dye that can be excited in the green range and emits very far out in the far red, so that you can hit it with that same 775 depletion laser. And that really has allowed to perform 3-color imaging, 3-color live cell STED imaging. And I am actually very excited about further development of these long stalk shift dyes in the next few years that will hopefully make even more than 2 colors STED imaging accessible to everybody. So coming back to our labeling strategy, you have your halo and SNAP tag, the proteins of interest, you express them in the cell, In this case, in this example here, we have SEC61, which labels the membranes of the endoplasmic reticulum, tagged with a halo tag, and OMP25, which is an outer mitochondrial protein tagged with a snap tag. And you simply add the dyes into your culture medium. These dyes are cell permeable, so are able to permeate inside the cells, and then they will react with snap and halo, and they will label your cells. And then you wash the cells and put them on your microscope. And back in 2016, we were able to show that this combination works with all sorts of different combinations of intracellular markers. For example, here in magenta, we have the endoplasmic reticulum, and in green, mitochondria with SNAP and halo and this dye combination. And we can see the hollow center of an ER tube, which is around actually only 100 nanometers membrane per membrane. And we can also catch events, for example, this mitochondrion down here is being constricted by the ER tube. So we also tested the same dye and tag combination with another sample, which consisted of clathrin coated pits labeled with the SNAP clathrin light chain construct, and an endocytic cargo. In this case, this is transferrin receptor with a halo tag. And we could see little donut-like structures, which are clathrin coated pits here at the plasma membrane. And in some cases, we could even see this clathrin coated bits filled with a transferrin receptor, like shown here in this still frame, which are being endocytosed inside the cells, like the one that I'm pointing at with my laser pointer down here. And with overexpressed protein, it's actually very easy to also do long-term dynamic, I mean long-term in the sense of like followed markers for over several minutes. In this case, we have labeled the lumen of the ER with the SNAP KDL. KDL is a tetrapeptide that keeps proteins into the endoplasmic reticulum lumen, and our membrane, again, the same membrane ER marker. And we can appreciate this trijunction, for example, where with the SEC61 marker, like kind of defining the outline of the ER, and the KDL filling the center, and also even these tubes, we see the center of the ER and the membranes. Actually, what's very interesting, like all these experiments were carried out with the previous iteration of a halo tag. However, I wanted to share with you that a few months ago, a preprint was published about a new halo tag version. It's called halo tag 9. Which apparently has improved brightness and increased fluorescent lifetime when labeled with some rhodamine, some commonly used rhodamine dyes. And we haven't tested this directly, but we are very excited about this development, and we actually have the plasmid in our drawer ready to be tested. So I would be really interested in knowing if anybody has compared with the older iteration of halo tag for lifestyle STED. Another thing that I wanna show you is that in our research we really need to label specific protein because we want to follow specific machinery components. However, if you want to label all the intracellular membrane, there is this beautiful tool that was developed by Marco Lamper at the MBL and Mike Heilemann's lab, where they use Nile-Red, which was previously used for single molecule localization microscopy, to actually perform lifestyle STED imaging experiment. And what Nile-Red does, it just labels all the intracellular membrane. You can clearly see ER tubules here and also mitochondria. And what is very important here is that this Nile-Red like comes on and off the membranes, providing like a very large reservoir of unbleached label. And I think this is also, I'm also really excited about further development of these probes, which hopefully in the direction of being able to selectively label various intracellular membranes. And So coming back to our biological topic, which is intracellular trafficking, I will try to show you some examples of what we do in the lab without going into too much biological detail. So I think one of the first important things that one person should ask is like, why do I need super resolution microscopy? Also, in particularly, why do I need to go through the trouble of doing live cell super resolution microscopy to investigate intracellular trafficking? So what is very important is to know, also for the non-cell biologists in the audience, is that while this is just represented like this as a scheme, all these organelles are all tightly packed in the perinuclear area of the cell. So perinuclear means the area next to the nucleus. And just to show you an example of the distances we're talking about, we're trying to understand how the Golgi intermediate compartment, which is this sorting station in between the R and the Golgi, is interacting with the neighboring organelle. And the Golgi intermediate compartment is only 100 nanometers or less away from the Golgi. Or the different pancakes of the Golgi stack are less than 20 nanometers away from each other and only 20 to 50 nanometers in size. So this gives you an idea of why we need the super resolution. I really like to show this image from Jennifer Lippico-Schwartz lab, because this really shows you what I mean. So this is the nucleus of the cell here, and this is the perinucleus, and all these organelles are all packed in the same area, and this area is very crowded. So if you really want to see how all these membranes interact, it is very important to have super resolution or super resolving power. And another thing that is very important for us and might also be important for other people in the audience is that some of these Golgi elements or also endosome elements are very tubular in nature, and the tubular endosomes and the vesicles are really sensitive to fixation. So when we fix the cells with paraformaldehyde or even like low concentration of glutaraldehyde, a lot of these structures completely disappear. So that's why it's very important for the live cell is very important for our research. And another thing is that we combine live cell STED together with gene editing tools. So we use CRISPR-Cas to tag our proteins of interest directly at the endogenous locus. And this is to avoid overexpression of these very important proteins. And I like to show this example because it is very striking. This is the small GTPase ARF1, and this is what happens when you overexpress this. You're just flooding the cytosol and you have a lot of background, and it's only when you gene edit, so you put the halo tag directly on the endogenous copy of ARF1, that you can really see structures and follow them. And what I want to do now, in the next 4 or 5 slides, I want to show you a couple of examples of what we are doing in the lab, and to hopefully convince you that this is a very powerful tool. So we are interested in how all these intracellular organelles communicate with each other. And even if you're not a cell biologist, you probably have heard from basic biology courses that communications between organisms is mediated by shuttling vesicles. So these vesicles, they form from a donor compartment and then fuse with an acceptor compartment. And we can finally, what I want to stress is that these vesicles are around 70 to 100 nanometers in size, depending from the vesicles, and are very close to the membranes where they form. So it is very hard to follow them with the standard diffraction limited light microscopy, but it is with using live cell STED that we can really follow the process of a vesicle forming from a membrane. And the examples that I'm showing you, this is clathrin light chain tagged at the endogenous locus with a halo tag. And this is clathrin that localizes to the Golgi. And you can see here from the still frames that we can finally follow a vesicle forming from the Golgi. Or we can also look at cluttering nanodomains, rearranging and restructuring on the Golgi, which is very important to understand how these vesicles are formed. And another very interesting topic, topic of research that we're pursuing from the lab is we're trying to understand how all these various endosomal compartments communicate with each other and exchange material. And there is beautiful work from a PhD student in the lab, Petya, which is focusing on really trying to understand how endosomal compartment exchange material and membranes with each other. And what she actually sees using live-cell STED is that different types of endosomes, like in this case, this is an early endosome labeled by RAB5 and recycling endosomes labeled by RAB11. These endosomes are kind of dancing around together. And I just want to stress that these structures are completely disrupted upon fixation. So we need to be able to do live-cell super resolution experiments. And another example here is from another PhD student in the lab, Louise. And what Louise is studying is how early secretory organelles exchange material. And again here, Louise sees these structures here, which are labeled by a small GTPase, which is called R4. And you can see that these structures are making transient contact with the other membrane. So, we think that that's how cargo transport is happening, by these transient tubular connections that are formed between the membranes. And if you think about this is the 2-micron scale bar, this is probably only like a few hundred nanometers again. And one last example here, we are also using live-cell STED to study the membrane organization of T-cell microvilli. Microvilli are these membrane protrusions in T cells, which have been shown to be very important for immune response and downstream signaling. And important signaling molecules, like for example the TCR, have been shown to be enriched in the tip of this microvilli structure. And what we can do now, we can endogenously tag some of these very important signaling molecules, and we can follow them live. This is work that was done by another PhD students and our Carmen and our technician Ellie. And you can see an example down here of this protrusion just kind of forming and then retracting again. And so this was my last example. And maybe I didn't get like a lot of the biology across, but I really hope that I convinced you that live-cell STED is really now allowing us to follow dynamic events at the nanoscale, like communication between organelles and membrane organization and localization, nanoscale localization of membrane proteins. And the live-cell imaging is really required for structures that are sensitive to fixation, for example, tubulovesicular organelles. And what live-cell STED is really allowing us here is to avoid using antibodies, which sometimes cause clustering artifacts, and also you lose resolution because of the use of the antibody. And we can really perform super resolution imaging in the native cellular environment, in particular when live-cell STED is combined with CRISPR-Cas9 for endogenous expression level. And I also wanted to end kind of with a more technical note. There is a few things that we have to consider, which is probably very important here for people that will want to perform these experiments in the future. There is things that we have to consider when choosing a red dye for 2-color live-cell STED. In particularly the crosstalk with the far red dye. These dyes are very close in spectra, so one has to be very careful that you, for example, your 640 excitation laser does not excite the red dye. The steadability, so if you pick a dye that is further out into the blue, you obviously have less crosstalk, so you solve the first problem, but then the problem is that you will get less resolution because it's because your dye it's less efficiently hit by the 775 STED laser. And also background issues that we're having. Halo substrate seems to be really performing better than the snap substrate. I think Florian maybe can explain you better if there's a question about that, better why that happens from the chemical point of view. And also choose your dyes based on the excitation laser of your setup, which is particularly efficient. Excitation is particularly important for endogenous expression levels, because you really want to hit, you really want to make the most of your dyes. So you really want to hit your dye exactly at the peak of excitation. And you also have to take into account that the red dye has a narrower detection window compared to the big detection window that you have for your far red dye. So I just wanted to leave this here, and I'm happy to discuss obviously I come into thank you notes. I am really, really lucky to have a wonderful lab, and in particular, I'll show you work from Petya, Louise, Carmen, and Ellie. And I'm always grateful to all the labs that share dyes and probes with us. In particular, I've shown you dyes that come from Vladimir Belov in the health department and the Luke Levis lab, and also my former colleagues at Yale that really allowed me to like fully develop these techniques while I was a postdoc there. And I thank you all for your attention, and I'm happy to take any questions.
Thank you, Francesca, for your nice talk, and there were really nice images and videos. So I really enjoyed it, and we got a couple of questions during your presentation. So I will start with the Q&A session. And the first question is, Francesca, is there any difference between the combination of dyes with either the snap or the halo tag? So have you ever experienced that one dye works better for one or the other tag?
I have a feeling that the halo is always less like sticky and always gives less background, and I think it's because the chloroquine versus the benzoguanine, and I think maybe a chemist might be able to answer this question a little bit more technically. But this is what we've noticed, and also other things we've noticed that some dyes work very well as like an actin or microtubule probe, but then absolutely don't work well for like as a snap. So I think you just really need to like try what works best for your application, and I'm obviously happy to answer questions also in my email if anybody has yeah.
Okay, yeah, thank you for your answer. So I haven't tried the halo so far, so I've just experienced with the snap tag. Another question, on average, how many frames can you record before you experience photo bleaching?
It really depends what you're looking at. Like, for example, if you're dealing with overexpression, you can, I would say you can go hundreds and hundreds of frames. But with endogenous expression level, it's always a little bit trickier, and you always have to find like the right compromise. So for example, sometimes we decide to image at like a slower frame rate to actually be able to catch like a longer time frame, because we cannot take as many frames. But we are lucky that some of the endogenously tagged proteins that we have are actually quite abundant. So I would say we can still take like 20, 30 frames before it's bleached. But for some other proteins that we have had CRISPR knock-ins that we couldn't absolutely see anything. So 0 frame confocal, 0 frame STED. But it depends. It depends how well expressed your protein is. Yeah.
Yeah, thank you. And another question, have you any, sorry,
I want to comment. I want to add something to the previous question about snap and halo. I think halo is much faster kinetic. Well, we actually have made a comparison, side-by-side comparison. So the same CRISPR knock-in background with snap and with halo And it's very obvious that the halo is much better in terms of like faster kinetics and labeling. We get like 10 times stronger labeling with the halo. So we always try to plan our experiments with obviously the crappier, less expressed protein on the snap, on the halo, so that we can crank up the signal, and the brightest on the snap, because we know that we can deal with less signals. So you always have to find the right balance. Yeah, sorry. Yeah.
No problem. Thank you very much. So another question is about background. Have you ever had problems with the background with snap or halo tag? And how do you minimize them, or do you have any tips and tricks there?
I have to admit that there's always quite a bit of background. In particular, you always see like little endosomal structure, but that usually is in the cells that are not expressed in your target. When your target is present, the background seems to be becoming much better. And again, comparison with snap and halo is that snap always gives much more of these problems than with the halo tag. So that if you have single color, I would always recommend to work with the halo. And yes, we don't have any solution for the problem. But the higher your target is expressed, and the less background issues you will have. So yeah, sometimes you just have to deal with the background. Yeah.
All right. Yeah, thank you for your answer. And I have one last question here for the moment. So have you also experienced with the halo or snap tag in fixed tissue or fixed cells? And is then one tag better for this approach, so for this application? Have you any experience with that?
Yeah, I have to admit that my experience was this, that when you fix the snap and the halo, the snap works better. But then I know people that have also worked with the fixed and then staining halo, and it works well in their hands. It never really worked. But I have to admit that now we're very careful. So when we make our knock-in, we want to try to avoid like fixing snap and halo and using antibodies. And what we do, we always attach epitope tags to our snap and halo so that we can use it for live cells and we can use it for fixed cells. We learned it the hard way. We generated some of initial CRISPR knock-in with halo, and then didn't work. And now we always attach small epitope tag to snap and halo. Yeah.
All right. Yes. Thank you very much for your answers and for your expertise here. So I have to say there are a lot of questions in the chat at the moment, so we will answer them later via email, but we have to move on to our second speaker. Francesca, thank you very much for being with us, for sharing your expertise. It was a pleasure, and thank you.
Thank you. Bye-bye.
And with this, I would like to introduce to you our second speaker today. So I have here with me my colleague, Florian Grimm. Florian is a chemist and biochemist by training, and he was actually one of the first employees at abberior. So he has a lot of background in the development and production of fluorophores and cell-permeable fluorophores, which are especially useful for live cell STED imaging. And he will share today with us his tips and tricks about live cell STED microscopy, and especially about how to keep your cells vital during the STED microscopy. So with this, I hand over to Florian. Florian, we are looking forward to your talk.
All right. So thanks, Julia, for the very kind introduction. And welcome everyone also from my side. And I'm very happy to talk with you about live cell nanoscopy. In the next minutes, I will talk a little bit about fluorophores, so the design of fluorophores, so what we actually need, that the fluorophore is cell permeable. I will also show you an overview about different labeling strategies, so what kind of tags we can use. And I will give some tricks and tips for imaging cells over a longer time period, so in time-lapse experiments. I mean, to really see dynamic processes in living cells, we can stain cells, of course, in different ways. In our case, we use organic dyes to see those dynamic processes, like in this example here, where we stained a membrane permeable, a membrane anchored protein in a confocal time-lapse experiment. And unfortunately, we are limited by diffraction. So we can also use those organic dyes for STED microscopy to observe the dynamic processes which are far below the diffraction limit. But what kind of organic dyes we can use? It turns out that in the last years, it turns out that rhodamines are actually one of the best suited fluorophores for those experiments. And here is an example of very schematical drawing of an rhodamine. So a rhodamine basically consists of a so-called xanthine ring, which is this three-membered ring system. And then beneath there's a benzolic ring, which with the carboxylic acid attached. And then on this benzolic ring, we can add any kind of ligands like reactive groups or functional groups. And with this, we create probes to very specifically stain target structures in living cells. And besides the fact that rhodamines, like other organic dyes, can really nicely or are very nicely or show a very high quantum yield and a high extinction coefficient, and are very good photostable, they exist also in two different forms. So the one I've shown you already is the so-called open form, where the fluorophore exists in the switterionic as a Switter ion. So, and also in this form, the fluorophore is fluorescent. However, because of this switterionic character, the fluorophore cannot enter the cell. But there's also equilibrium, so that the fluorophore can exist in this so-called closed form. Means that this negatively charged oxygen here can interact with the center carbon of the xanthine ring to form the so-called lacton ring structure, or lacton ring. So the fluorophore now is non-fluorescent, but also non-charged, and can therefore easily enter the cell by passing the membrane. And once the fluorophore has reached the intracellular space, it can also exist in these two forms. And with a ligand, it will find the target and can interact or react with the target very specifically. So now it is quite interesting to understand the equilibrium between the open and the closed form. And I will show you, or I will show you why it is so important. So the fluorophore, basically in a polar environment, the rhodamine is in a polar environment in this open form, in the fluorescent form, but in an unpolar environment, the equilibrium will shift to the closed form, to this lacton ring structure. So in this movie, you see a reactive tube, and beneath this tube, there's a UV lamp. And in this tube is filled dioxane. Dioxane is a very, very unpolar solvent. And in this dioxane, we have dissolved rhodamine. And because we are in an unpolar environment, the fluorophore is in this closed non-fluorescent form. But once we will shift, or once we will come from an unpolar to a polar environment by adding water to this tube, the fluorophore, or the equilibrium, will shift to the polar, to the open form, and we see this fluorescent signal. So now we, or how we can play around with this equilibrium. So how can we create fluorophores being either in this fluorescent state, but also have the possibility to form this non-charged form, this closed form. I will show you this in two examples. So let's have a look on TMR, tetramethyl rhodamine, which is a very famous and a very classic rhodamine. The emission wavelength, or the emission maxima of this rhodamine, is at 570, 580 nanometers, but the permeability is quite low. Means that it is not that likely that this fluorophore can also exist in this closed form, or can form this form this lactone ring. But we can exchange a lot of things on the basic structure of a rhodamine. So we can add groups, functional groups to our groups to it. We can exchange atoms in this rhodamine. So here's the first example. So the so-called Life 510, where we add this fluorine atoms here on the xanthine ring system, which are electronic withdrawing groups. This has two effects. One effect is that we shift the spectra into the blue region. So now the absorption, or the emission maxima, is at 509 nanometers, and the permeability decreased drastically. The reason here is that we decrease the electronic density in the xanthine ring system, so that the center carbon here, the electrophilic character of the center carbon, increases, and therefore it is easier for this fluorophore to form this lactone ring. Another example, if we return to TMR, we can exchange this oxygen by another atom, for example, by a carbon. Now we, or that is a new class of fluorophore. It is not a rhodamine anymore. It is a so-called carbopyranine, and we named it Life 610. And there are also these two effects. One is that the spectrum will shift into the red region, means that the fluorescent maximum is now at 620, 625 nanometers, and the permeability will increase also. The reason is the same, basically, because we destabilize the positive charge, and therefore the fluorophore can, or it is easier for the fluorophore to form this lactone ring. Now, what else we do need for good fluorophores being suitable for life cells? STED microscopy. Well, the spectra has to fit with our STED STED laser. This is a slide Francesca have shown us already. So briefly, we need an excitation laser, of course, to excite our fluorophore. In this case, it's a 640 nanometer laser. We have our detection window, and then also our STED laser. And this STED laser has to overlap with our or with the spectra, with the fluorescent spectra of our dye for stimulated emission. So we can add another fluorophore, which also fits, so that we can excite it with a second laser. So we have then a second detection window, and it can be also used with a STED laser. Okay, so now after creating or after having all these things in mind, we can create different types of different fluorophores covering more or less the spectral range. So we have the Life 510, which can be excited with 488 nanometer and can be used with the 595 STED laser. And in the orange or yellow, orange, and red region, we have this fluorophore called Life 550, 560, 590, and 610, which can be excited either with the 561 nanometer laser or with the 640 nanometer laser, and can be also used with the 775 nanometer STED laser. So having all these fluorophores, we now need ligands to really like tell the fluorophore where to go. And now I will give you a brief overview what kind of tags we can use right now for our fluorophores. So one example, Francesca have spoken a lot about it, so I can really go briefly over this slide. So it's like the self-labeling proteins like SNAP-tag, for example, where we have our plasmid with a gene of the protein of interest and the gene with our protein tag. And the cell will transcript and translate this to our fusion protein, and from the outside we will bring the flu force into the cell. And here's the reaction. So we have this reactive group, so a benzyl guanine on our fluorophore, and this self-labeling protein will react with this benzyl guanine, releasing the guanine, and the fluorophore is covalently bound over thioether to our fusion protein. Another ligands are those small molecules I've shown you here. So one is Höchst. Höchst is an DNA binder, so it will interact with double-stranded DNA. And when we bind the Höchst moiety on our fluorophore, we can nicely create DNA probes, which very specifically, specifically stain the DNA. Another example are this peptide here, the cyclic peptide, the so-called jasplakinolide, which interact with effectin. And also here we place a fluorophore to this peptide and can stain nicely the actin filaments and the actin network in living cells. And also taxols like carboxytaxel or laotaxel or docetaxel will interact with beta tubulins. And also with a fluorophore attached, we can create tubulin probes staining specifically the tubulin filaments in living cells. And if you are interested in those small molecules, you should join us on our next online symposium, which is on the 15th of September. And Grashvidas from the Max Planck Institute here in Göttingen will give us a more detailed overview about those small molecules and probes. And the third example are tetrazine and azides as reactive groups. So what we do with this is that we have this unnatural amino acids. So here are the two examples, this modified lysines, where we have this cyclic octene or octane rings, or cyclic heptane or heptene rings. And we use this unnatural amino acids to replace any kind of natural amino acids in our protein of interest with a modified tRNA to build in these unnatural amino acids along our protein of interest. And with this, we can create a bioorthogonal reactive group, which will react without adding any kind of reagencies, will react with tetrazine or azides to bind covalently on our protein of interest. And then we have labeled specifically our protein of interest only with one fluorophore and with a very, very small tag. So now, after talking about also, if you're interested in this kind of labeling technique, Ivana will also give us a talk on our next online symposium, which is also on the 15th of September. So if you're interested in this labeling technique, please join us after the summer break. So now, after having the fluorophores and knowing the tags and combining everything to nice fluorescent probes, I will show you now some examples, some images, and also give you some tricks and tips how to image living cells with the STED microscopy technique. So here is a mammalian cell stained with live 610 tubulin. You see very nicely that is a STED image, and all other images I will show you now are raw data. So you see here very nicely this single filaments, and if you look or if you have a look to this close up here, there is this confocal image on the very on the lower left, and the STED image on the upper right part, and you very nicely see the difference between confocal and STED. And if you draw a line profile over this filaments here, you see in red that you can distinguish between each and every single filament by STED microscopy, and in gray, that is a line profile over the confocal image, and here you cannot really distinguish between the single filaments. Another example is the Life 590 DNA probe. Here I will show you the dynamic processes of a dividing cell. So you can observe the cell over more than an hour without bleaching and seeing this deviation. We also can use our fluorophores in combination to create two color images. Here we used the construct which was a gift from Fran, the outer membrane protein OM25, which will stain a protein inside the mitochondria. And we used the molecular probe 550 tubulin to stain the tubulin structure here in green. So the upper one is the confocal, and the lower image is a STED image. So you see that this fluorophores can be used in combination. And also we are working hard to provide you with a third color, with a third fluorophore suitable for live cell STED microscopy. What you see here is a mammalian cell stained with Live 610 actin, Live 560 tubulin, and with our hopefully new dye labeled with the benzyl guanine to stain a SNAP construct. So here in this close-up, you see the actin filament, the tubulin filament, and the endoplasmatic reticulum, the ER, stained with these three fluorophores with a STED microscopy. That is the overlay. And if you have already a guess what this third color can be, you can write us. You can tell us your guess. And if you're guessing right, we will provide you with a little goodie. Okay, now let's talk about some important settings you have to always keep in mind by live cell STED microscopy. So if you look to this image here, that is a confocal image where we used 0 STED power, then you can, of course, easily observe the tubulin filaments over a longer time. This has been, or we have imaged every minute 1 frame for half an hour. So we imaged mammalian cells and stained them with our probe Live 610 tubulin. Now what will happen if we increase a little bit the STED power? Of course, we will achieve a little higher resolution, like with 5%. And we can still observe our filament easily over the same time period. So that is, of course, not the best resolution we can achieve. So we have to increase the STED power even further to get an increase in the resolution. And with 20% STED, we can still observe these filaments without bleaching effects or destroying the structures. Keep in mind that increasing the STED power does not mean that your resolution will increase linearly. You see this in this curve here. So with 20%, we have also already reached mostly the best resolution we can. So increasing it even further means for higher resolution with like 5 or 10% better, we have to increase the STED power even harder and even further. And this brings us to a point where the fluorophore might be bleached, or the structure will be destroyed. So this is what you see here. The resolution is quite good, but bleaching will start after a few frames. Another example of important settings is the pixel size. So of course, You can decrease the pixel size to 20, 50 nanometers to really achieve good a good quality of your image. It's like the same with a camera. If you have a 3 megapixel camera, then the quality is quite good. If you have a 10 megapixel camera, then the quality is even better. But what will happen if we have a very, very small pixel size? So that is a fluorophore, and each area here is 1 pixel. And STED is the same like with confocal. We're having a scanning microscopy technique, means that we start from the very upper left to the lower right pixel by scanning our region of interest. Now, this fluorophore, or what will happen with the fluorophore when we start scanning? So it will stay quite a long time in the excitation spot and then also in our STED spot. But if we decrease or if you enlarge the pixel size, then the fluorophore will stay shorter in this excitation spot, and quite a short time if we enlarge the pixel size even further. Now, to show you this on an example, we stained mammalian cells again with Life 610 tubulin, and we keep everything equal except of the pixel size. So that's 20 nanometer, 30, and 40 nanometer. With 20 nanometer, you see a very, very good quality of your image. But what will happen when we scan like 30 times over the same region of interest? Then we see bleaching, we see that our structures are getting destroyed, and we also see that the cells are dying. Now, if we use the same setting with 30 nanometer pixel size, then we can observe in the same time period the cell much nicer. So there's a little bit bleaching for sure, but the structures are fine and the cells are alive. And with 40 nanometer pixel size, the quality of your image is rather bad. But the cells are healthy, the structures are good, and we do not see any bleaching. So to summarize this, what you should keep in mind is that with a STED power, use as low STED power as possible, optimize the pixel size, and for fluorophores, it is often quite good to lower the dwell time, but increase the line accumulation. And if you have very tricky samples, if you have very tricky proteins you are going to image, you want to observe, then another very good technique is the adaptive elimination, which is a powerful tool which can be used. But all in all, be gentle to your cell, to the target, and of course to your fluorophore. And then you're having fun with live cell microscopy for sure. So with this, I will thank you for your attention. And if you have questions, go to our website, abberior.com. There are a lot of information about fluorophores and the microscopes. And if you're not finding your information you're looking for, then write us a mail or give us a call, and we are always happy to help you. But for now, I would like to take all your questions.
Thank you, Florian, for your talk as well, and for providing your tips and tricks for life-size STED microscopy and how to keep these cells alive and still get a good resolution. And we also got a lot of questions during your presentation, so I will also start with the Q&A session. And the first question is, on average, how long can you image your cells?
Well, it is of course different depending on the cell type, of course, and it depends also on the structure. Like with tubulin, it is no problem to image the structure for hours. But if you have not so density or dense packed stainings, then of course it's a little bit more complicated. and yeah, it's a little bit less in time, and it also depends how often you're scanning over your frames.
Thank you for your answer. So it depends on several factors. Another question is regarding your staining protocol. So can you give some information about the concentrations you use for staining and how long you stain? So for example, for the tubulin or DNA probes?
We use normally a very, very low concentration for our probes, or with our probes to stain the cells. For tubulin, we basically use like 0.1 micromolar to 10 nanomolar concentration, which is very, very low, because these probes can very nicely go through the membrane, and they are very, very specific for these and have a high are very specific for this tubulin and DNA probes, and as well for actin. So we use basically a very low concentration, like in the nanomolar range, 10 to 100 nanomolar. Yeah.
Thank you for sharing these informations. And another question: are the live dyes suitable for different cell lines? And have you tested different ones? Can you elaborate a bit on that?
Yes, we have tested a few cell lines. Of course, not all cell lines which are available, but the standard cell types we have tested, like fibroblasts or other cell types. And we have also tested it on tissues. So we have tested it on Drosophila, which is also working quite nicely. And also there, the background is very low. So it's working in tissue as well. Yeah.
Also regarding a bit to testing different cell lines. So do you have any experience with toxicity in cells, and have you tested that?
Yes, so the toxicity is, of course, especially with these small molecules, like with tubulin or actin, a problem. Because tubulin, for example, taxols are toxins which are used in cancer treatment, right? So a very important thing is to use as low probe as possible. Means what you probably could do is to increase the staining time, let's say overnight, like 12-16 hours something, but decrease the amount of your probe by a factor of 10 or 20, and then the quality of staining is quite the same, but the cells are more healthier. That is something we have figured out here in-house, and it's also in-house, and it's quite a good trick. Yeah.
Thank you for these answers. So I guess toxicity is of course an important topic. And another question is: can you use the DNA probes, for example, also in fixed cells and in fixed tissues?
Yes, those probes can be used, or I mean, the actin probe and the DNA probes can be used in fixed cells and also in fixed tissues. Tubulin, however, is not suitable for fixed cells, but these two, actin and tubulin, can be used in fixed cells and tissues, yes.
So that's a really cool thing and can be an additional option because of antibody specificity. Okay, yes, thank you. So thank you, Florian, for your nice talk, for answering the questions. And with this, we are at the end of our today's session. And of course, I would like to thank you as well for joining us. And if you got now interested in our live dyes or in our microscopes, please contact us, and we are happy to answer questions or to demonstrate our dyes or microscopes to you. And we now do a small summer break, and our next session will be in September, and actually it will be the follow-up session for today, but a bit more about labeling techniques. So we hope to see you back in September. Stay tuned on our social media channels, and with that, stay healthy, and goodbye.